Well, good morning, everybody. I think that it's time to start our lecture. In according to our tradition, please remind me what is the first step of the development of a neuron? What is the first step of the development? Do you remember the schematic representation of the development of the neuron and of the neuron and of the brain? Yes.
Stem cell proliferation and differentiation.
A little bit louder, that's for everybody.
Is the first step the proliferation of stem cells and their differentiation?
Yes. Now, the proliferation should be ceased, and the proliferation of what cells? What is how they are called?
Stem cells?
Stem cells. Okay. Well, and after the cessation of the proliferation, the neurons appear, non-differentiated neurons. And the second step,
Their migration to the place of their functioning.
Exactly. Well, and at the same time, there is an expression of all specific chemical synthesis. And of what kind of molecules? When I say specific synthesis, what do I mean? Well, yes, molecules. Then, what is the most important group of neurotransmitters? The largest group, neuropeptides. Okay, monoamines. Good. Okay, and now let's continue and let's finish this topic: development of the brain and the regulation of the brain development and brain differentiation. Well, so. Let's go to the point which is called regulation of neuron development. Now, so this is a schematic representation of the neuron development, of the establishment of interneuronal relations, okay? And it should be mentioned that at any step of the development of the brain, there is a control of the development by physiological active cells, by neuropeptides, or classical neurotransmitters, monoamines, acetylcholine, etc., etc. At any stage, neurogenesis, neuron migration, development, synaptogenesis, etc. Well, and there is a high group of chemical signals which control the development of the brain at any step of its development. These are growth factors. Growth factors, so these are small neuropeptides, which, what is the role of growth factors? I repeatedly mentioned. What is the role of growth factors or neurotropic factors? They serve as neuroprotectors, on the one hand, and they promote the development of the brain and the differentiation of the neurons. Also, one of the most representative growth factors is insulin-like growth factors type 1. Also, it promotes proliferation of neural precursor cells. How do we call neural precursors? Nobody knows except you. What is the title of the progenitor cells for neurons?
Stem cell.
It was mentioned 10 minutes ago.
Stem cells.
Stem cells. Good. Well, and then osteogenesis and oligodendrogenesis. Well, and how these cells are called astroglial cells and oligodendrocytes. What is the type? Are these neurons or other types?
Glial.
Glial cells.
They're glial cells, yeah? Glial cells. And what is the third type of glial cells, which is not mentioned here? Sorry?
Microglia.
Microglia. Yes, exactly. Well, then the neurons surviving, because you remember that during the perinatal period there is a great apoptosis of neurons, because neurons are produced in too much high, high level. Then exogenesis and exoguidance. And the establishment of synaptic interrelations between differentiated neurons. Well, so all these steps of the neuron differentiation and of the glial development are under the control by growth factors, not only by insulin-like growth factors. There is a number of others which are very important. Well, and now let's turn to the sexual differentiation of the developing brain. Today at our seminar we will have two presentations on the masculinization, so-called masculinization of sexual differentiation of the brain, and therefore I will give you this information very schematically. Well, so what is most important? That there is sexual dimorphism in the brain of adult humans. Well, so men have heavier brain than women. Women's brain have a high glucose utilization. What does it mean from the physiological point of view if there is a high level of glucose utilization? Glucose is a substrate for what? What kind of processes?
For energy.
For general metabolism. Eh? Well, so the general metabolism in the neurons in women is higher than, well, so they are more, these neurons, they are more active, they intensely functioning, etc. Glucose utilization and the weight utilization, the weight and the blood flow. And then this sexual dimorphism is manifested, as it has been shown here, in the number of neurons, in the length of dendrites, the number and the amount of synapses, the number of synapses. Okay? And there is a great concentration of estrogen receptors. Yeah, please. Very good. Well, you are listening. Well, so there was a. What? Excuse me? Okay. Well, so there is in adulthood there is a great concentration of receptors for estrogens. Estrogens are gonadal hormones of women, and you can see that there is a high concentration of these receptors, particularly in the hypothalamic region, which is responsible for. The hypothalamic region is responsible for what? Neuroendocrine regulations. Good. And well, and also in the midbrain and in some other regions. In the midbrain, the estrogen receptors are involved in the regulation of sexual behavior. Well, and now let's try to understand what is the mechanism of the sexual differentiation or masculinization of the brain. I mentioned it already during our previous lecture that just after birth, the brain in the females and in the males is the same, absolutely the same, in structure and in function, just after birth. But then the brain of males is converted, is reorganized, and in structural fashion and also in the functional fashion. Well, so particularly the males, the brain of the males control male behavior. Well, so, and what is what is surprising that the muscularization of the brain is realized under the control not by testosterone directly. Testosterone, this is a hormone of males, huh? But by estrogens, which are the hormones of females, sexual steroid of females. This is a paradoxical reality, huh? Well, how we could explain it? Look, please, there is newborn males and females. There is a secretion by gametes of estrogens in females and testosterone in males. And estrogen is responsible for the muscularization of the brain. But this is not the case for females, because estrogens, which are delivered into the bloodstream, they cannot reach the brain. You understand? But for what reason? I mentioned it once, but that was, I think, at the first lecture. Why estrogen, which is circulated in the bloodstream, why it cannot reach the brain in females?
Blood-brain barrier.
No, for steroids, I mentioned several times. For steroids, there is no blood-brain barrier at all. But what is the reason? Because in females, during this period of the brain muscularization, there is a very high level of circulating alpha-fetoprotein. This is a special protein which is linked to estrogens. No? So it binds estrogens in the bloodstream, and in this complex, it cannot be delivered from the bloodstream to the brain. Huh? Understand? Well, but on the other hand, in males, the brain is muscularized by estrogens. How it happens? Well, so in males, there is a secretion of testosterone into the bloodstream. This is a hormone of males, the sexual hormone of males. And testosterone is capable to enter the brain. Is capable to enter the brain. Huh? But in the brain, it is converted by an enzyme which is called aromatase. It is converted to estrogens. I repeat once again. First, there is a secretion of testosterone into the bloodstream in males. Then testosterone reaches, is coming to the brain. There is no blood barrier, etc. And in the brain, testosterone is converted by aromatase enzyme, aromatase, into estrogens. And these estrogens are responsible for the muscularization of the brain. Is it clear? Yeah, please.
So it is converted. Testosterone are converted to estrogens.
Yeah.
Is this estrogens the same as the estrogens in females that initially are not able to penetrate into the brain?
Yes. Well, so estrogens are the same in females and in males. But in females, estrogens are secreted to the bloodstream, and they are bound to the great protein, which is called alpha-fetoprotein. And in this complex, estrogen alpha-fetoprotein, this complex is not capable to penetrate into the brain.
Okay. So what if there is no binding with the alpha?
Yes, but in males, there is no alpha-fetoprotein in the blood over the spirit, in the brain, on the one hand. And on the other hand, alpha-fetoprotein does not link to testosterone. Okay. Yeah. Understand? Good. Well, and it's in fact it is well. So this is the best example of the control of the brain development and of the neural differentiation by the so-called epigenetic factors. In fact, we are coming to a general conclusion that there is a genetic program of the development of the brain and of the differentiation of the neurons, but it is flexible and it is under the control. It can be modified by so-called epigenetic factors, factors which are exterior vis-a-vis the neurons. Okay, and sexual hormones. This is an example. This is an example of epigenetic, one of the epigenetic factors. Okay. Yeah, please.
So, if we put a lot of male hormones to the male in her childhood, it will make her brain like male brain. We give her a lot of male hormones.
Testosterone. Yes, yes, exactly. Well, so on the contrary, for instance, well, so testosterone and estrogen, these are two sexual hormones, huh? But they are secreted both in females and males. But in males, the level of secretion of testosterone is significantly higher, multiply higher than in females. And on the contrary, the estrogen secretion in males is significantly less important than in females. Well, so it could happen during the abnormal development that there is a high concentration of testosterone in females. And in this case, the brain of the females will be converted to the brain of males. You understand? Well, so and these are real diseases, and they are called in general, they are called congenital diseases. Congenital diseases which are developed because of the impaired metabolism of these epigenetic factors during the critical period of the development of the brain or other organs. Understand? Good. Well, well, so that everything is right here. I will not repeat it again, once again. Well, so in the impairment of sexual differentiation, well, so we discussed already a little bit, and we will discuss more precisely at our seminar that inhibition of aromatase in males during critical period of muscularization results in showing female sexual responses, lordosis or receptive behavior in animals, retaining of male sexual activity with receptive female. The above features correspond with those found in transgender men. Well, so I repeat once again that very often I listen to the information which is coming from TV or from media, from newspapers, that there is a lot of transgenders in the United States, for instance, and this is a manifestation of the degradation of society, of moral degradation, etc. But I never had, probably you succeeded to hear it, but I never heard that this is this can be, this is a disease. In most, yeah, I have no idea to say that in 100% of cases, but in certain cases, this is a real disease. I've never heard about it, unfortunately. Well, human sexual irritation is determined by changes in the endocrine environment during early development, perinatal development, in fact, and in effect of epigenetic factors. Well, let's go on. It's clear. Well, now let's come to the general conclusion for the development of the brain and the molecules and of the neurons. Brain-derived signal molecules contribute to multiplied and parallel modulation of the neuron development in critical period of the genesis, mostly before the establishment of synaptic interneural relations and the blood-brain barrier. Specifically, monoamines inhibit the proliferation of progenitor cells, accelerating neurogenesis. They also provide tonic inhibitor impact on the differentiating serotoninergic neurons of the rafting nucleus via autoreceptors, stimulate migration of the differentiating target peptidergic neurons toward the place of their final settling, provide the short-term impact on differentiating peptidergic and monoaminergic neurons during the intrauterine development of inhibiting the neuropeptide expression in the former and stimulating the tyrosine hydroxylase expression in the latter. Also, tyrosine hydroxylase, what is the functioning of this enzyme? Tyrosine hydroxylase. This is an enzyme which is responsible for what reaction? What is its function? I repeated it dozens of times.
Is it involved in synthesis of L-tyrosine to
Exactly. This is the first enzyme of catecholamine synthesis. All catecholamine, dopamine, noradrenaline, adrenaline, norepinephrine, epinephrine. Well, so it converts L-tyrosine, the amino acid precursor, to a dopamine. Okay, the needed precursor of dopamine. Good. Well, provide long-lasting effect. Well, what is important? Provide the long-lasting effect on peptidergic neurons during the intrauterine development that is manifested by the attenuation of the neuropeptide synthesis release in adulthood. Do you remember that I mentioned that there are two fundamentally different actions for the same chemical signals to the same receptors? What is the fundamental difference between this action in adulthood and during the development of the brain? Please, come on.
In the developing brain, this action is long-lasting and it affects the
Effervescence.
gene expression.
Yeah,
And in adulthood, this is a more short-lasting effect.
exactly. Well, so in adulthood, this is an activating effect for a short period, just during the association of a chemical signal with the receptors. And you are quite right that during the development, this is a short-term, long-lasting effect, which is irreversible, in fact. You would like to say something?
I'm getting a little confused with point number 3 or 2, inhibition of stem cells.
This?
Yes.
Yeah, okay.
So I don't know whether it's my English that is not working. I don't understand that point. That
You don't understand what does it mean inhibit the protein?
yeah, when monoamines inhibit proliferation of stem cells, it accelerates neurogenesis.
Well, so, look, I'll repeat again. Well, so, the neurons and glial cells are generated from progenitor cells, which are called stem cells. These stem cells are included into the wall of the ventricles, so the cerebral ventricles, and they always proliferate. They are passing mitotic divisions. But at a certain period, under the control by chemical signals, the proliferation of the mitotic division is stopped. And from this period onwards, there is a development of the neuron. So this is the first step of the appearance of the neurons, of the origin of the neurons, just after the scission of mitotic divisions. It's not clear still?
So I get you right. If there's still migration of stem cells to their final destinations.
Well, so look, there is no migration of stem cells. After the appearance of the neuron from the stem cells, the differentiating neurons, which are not mitotically divided anymore, they migrate. The lowly differentiated neurons, they migrate along the basal processes of network cells to the places of their final settling in the brain. Would you like me to draw it?
No. I remember the drawing
Let's
last
go
time.
ahead. Good. Well, so the developing brain undergoes sexual differentiation during a critical period of ontogenesis under the influence of sex hormones. And now let's go to another lecture. These are brain diseases. Etiology, well, so before we discussed the brain, structure and functioning. We discussed the control of the brain, the development of the brain, and now we will turn to brain diseases. Etiology, this means, do you know what does it mean, etiology?
The origin.
Because it's the
Yeah, let's really look at the causes, yeah? What are the causes of which promote the development of the disease? Pathogenesis, these are cellular and molecular mechanism, mostly cellular and molecular mechanism of the development of the disease, diagnosis, and treatment. Okay? Well, and let's say that in the most often neurological and mental disorders are related to, are caused by the neural death. Well, so under the neural death, there is a deficiency of certain functions which are under the control by these neurons or their networks. Well, so this and all the diseases, they can be divided into two types: acute diseases, trauma, stroke, and chronic diseases, neurodegenerative disease. Chronic, it means that they are developed for a very long time, and acute, that they appear immediately, like trauma after stroke, etc. Well, but there are also the examples of brain diseases which are developed under the functional stimulation of the neurons, not because of the degradation of the neurons, but on the contrary, by the activation of the neurons. Do you know this?
Epilepsy.
There are only a few of them, huh?
Epilepsy.
Epilepsy. No, I'm not sure. I don't know exactly, but no, I'm not sure because in epilepsy there is a degradation. As far as I know, I'm not really a specialist, but this is schizophrenia. Schizophrenia because there is an activation of dopaminergic neurons. There's one of the hypotheses. It is called dopamine hypothesis of the development of schizophrenia. It is developed due to the activation of dopaminergic system and on the excess of dopamine. But this is really an exception. Well, and I repeat once again that there are two types. There are two types of the neuron death, which are fundamentally different. This is necrosis. The neurons are degenerated according to necrosis at acute diseases, trauma and strokes, and apoptosis. Apoptosis, it means that this is a genetically predetermined disease of the neuron, and it is developed under in the chronic diseases like Parkinson's disease, like Alzheimer's disease. Okay. And now we could differentiate, we could precise several types, several elements of the pathogenesis of neurodegenerative diseases. This is the neurodegeneration, the death of specific neurons. This is prion-like distribution of toxic proteins. What does it mean? It means that in some diseases, like Parkinson's disease or Alzheimer's disease, there is a production in some neurons, there is a production of toxic proteins, like aggregated alpha-synuclein or beta-amyloid in Alzheimer's disease. Well, sure, there is a production in individual neurons. But these toxic proteins are released from these neurons, and they can be captured by the neighboring neurons, by the next neurons. In this conglomerates of toxic proteins provoke the degeneration of these healthy neurons. Understand? Well, so one neuron which produces toxic proteins like aggregated alpha-synuclein, like beta-amyloid, these toxic proteins is released out from the neuron, and it is captured by a healthy neuron, thereby provoking the degeneration of these healthy neurons. This is called prion-like distribution of toxic processes. Well, so then there is another element of the pathogenesis of neurodegenerative diseases. This is neuroplasticity. Due to neuroplasticity, the diseases like Parkinson's disease and Alzheimer's disease, they are developed for decades without any manifestations of clinical symptoms. What kind of, could you remind me at least a couple of mechanisms or manifestations of neuroplasticity at the cellular level and at the molecular level? Okay, good.
I don't know, maybe let me try something. Is it one of the mechanisms where at the cellular level, where say a break in the axon, the process of a neuron gets regenerated in multiple processes?
Excellent. There is organization of damaged actions, organization with the appearance of a lot of axonal terminals. Well, good. That's one of them. Then there are, for instance, the replacement of degenerated neurons by the newborn neurons, which appear from stem cells, for instance. Yeah? Okay? Good. Excellent. And a couple of mechanisms, molecular mechanisms of neuroplasticity. Could you? Yeah, please.
As far as I remember, the synthesis of neurotransmitters, of different neurotransmitters, to adapt to the lack necessary neurotransmitters, and also changing the sensitivity.
Yeah, absolutely. Well, so the set of neurotransmitters in the same neurons can be changed, right? In different functional state of the brain on the one hand, and on the other hand, the sensitivity of the neurons for these neurotransmitters can be also changed according to the level of the neurotransmitters in the extracellular space, in the synaptic space. Well, so that's quite right. Well, now let's go to the prion-like and stimulation of separation of growth factor. So there are unspecific mechanisms providing neuroprotection, stimulation of secretion of the growth factors, and the activation of the antioxidant system. And specific mechanism of neuroplasticity to maintain neurotransmitter stimulation of neurotransmitter secretion, prevention of a loss of neurotransmitters in the extracellular space. Look, my question is, how is realized these mechanisms in the case of the functioning of a dopaminergic neuron? Well, so prevention of a loss of neurotransmitter in the extracellular space. Well, so if we are saying about dopaminergic neurons, it means that the neurotransmitter is dopamine. Huh? And what are the mechanisms of prevention of a loss of dopamine in the extracellular space? There are two mechanisms at least. Huh? Yeah?
If it is less of dopamine in intercellular space, some cells, still alive cells, can be more sensitive for it.
No, that's another story. This is a story about the role of dopamine in the regulation of sensitivity or the number of receptors for dopamine.
Yeah.
But I mean another way. Well, so after dopamine release into the intercellular cleft, and after its action, functional action, it should be eliminated from the intercellular cleft. And there are two mechanisms for the elimination of dopamine from the Good. The first mechanism is the uptake of dopamine by specific dopamine membrane transporter. Well, and another mechanism. This is a specific mechanism for any monoaminergic neurons, because the monoamine transporters, they exist in all monoaminergic neurons. In noraminergic neurons, norepinephrinergic neurons, norepinephrine transporter. In dopaminergic neurons, dopamine transporter, and they are specific. And there is a non-specific mechanism for the elimination of all the monoamines.
Monoamine oxidase.
Monoamine oxidase, exactly. The enzymes for degrading of monoamines. Good. Increase of the sensitivity of the target neurons. Well, so you told us already that there is, at least in dopaminergic neurotransmission, there is a sort of upregulation and the decrease of the amount of dopamine in the extracellular space. There is an increase of the number of receptors for dopamine on the target neurons. Okay. Good. Now let's turn to the classification of neurological and mental disease. There is a lot of classification all over the world, and you don't need really to know into all of them, but you should learn just the principle of classification. Classification of neurological diseases. First, disorders caused by damage of the local brain areas, affecting brain functions such as the speech, language, movement, memory, and cognitive skills. Well, the damage, what are usually the reasons for the damage? What causes produce the damage of the brain? Stroke. Stroke, and another one I mentioned before. Trauma. Yeah, trauma and stroke. Okay. So it depends on the functional consequences, the pathological consequences of this stroke or trauma is strictly depends on the region, on the affected region. Then disorders affecting the spinal cord, which is caused by injury, inflammation and other factors, which is caused by injury. Okay? Disorders affecting the peripheral nervous system, autonomic nervous system disorders, seizure disorders, movement disorders. What is the most important movement disorder? Do you know? At any lecture, I repeated dozens of times. I call this what? Parkinson's disease, certainly. Certainly.
This, so what about the other side of it, the other side of the question?
Other diseases.
Huntington's.
Huntington disease, yes. Huntington disease, yes, yes. Not typical Parkinson disease, which is not related to the damage of dopaminergic neurons and the nervous system, etc. But Parkinson disease is certainly the most important, the most widely distributed, and the best target disease. Well, then sleep disorders. So sleep disorders, they are developed because of the damage of what region of the brain? Suprachiasmatic nucleus. Good. Suprachiasmatic nucleus is a biological clock for the whole body. Huh? Interesting. Then, certain types of headaches, such as migraine, tension headaches, cerebrovascular attack, and stroke, demyelinization effect, such as multiple sclerosis, tumors affecting the nervous system, head injuries, infections affecting the nervous system. Well, do you know what is the same initial symptom, clinical symptom, in Parkinson's disease, in Alzheimer's disease, and in COVID-treated patients? There is the same first manifestation.
Loss of,
Sorry?
the loss of action.
Exactly. Exactly. That's all.
The loss
The-
of what?
Smell.
Oh,
Well, so and this is very natural because all three diseases are provoked by at least some of patients, are treated by exogenous toxic factors, like pesticides in Parkinson's disease and COVID viruses, and they entered the brain while the nerves bypassing the blood-brain barrier. And therefore, the first region, the initial region of the brain, which is touched, which is affected by these factors, are olfactory lobes, which are responsible for smell. Well, and let's go back. Then classification of mental diseases. Neurodevelopmental disorders cover a wide range of problems that usually begin in infancy, childhood, or the teenage age. Autism, attention deficit, and learning disorders. Some of them are genetically predetermined disorders, but some of them are congenital diseases, which are related due to the impaired metabolism of the morphogenetic factors during the critical period of the development of the brain. Then schizophrenia, spectrum and other psychotic disorders, bipolar and related disorders, depressive disorders, anxiety disorders, trauma and stressor related disorders, dissociative disorders, sleep-wake disorders, and neurocognitive disorders. Neurocognitive disorders, certainly the most representative is Alzheimer's disease. Well, so I mentioned already. Yeah, please.
Just motor neurons disease, just it's part of movement disorders.
What?
Motor neurons disease.
Motor neuron disease. Well, so-
Neurological movement disorder.
Yeah, that's true. The motor neuron disease. These are the neurons which innervate directly the muscle cells. Yeah, you mean? Well, so muscle cells, and there is a pathology which is related to the impaired synaptic-like contacts between the axons of these neurons and the muscle cells, okay, which are responsible for the movement. Good. And now let's turn to precisely to neurodegenerative diseases. We're saying about current insight in the pathogenesis, diagnosis, and treatment. I will tell you mostly about Parkinson's disease and Alzheimer's disease. And I should say that there is the same scenario in the development of these diseases, but these diseases provoked the damage of different functions. Alzheimer's disease, memory, learning, social adaptation. Parkinson's disease, movement, yes. Okay, good. Good.
Well, I think that in Parkinson.
Sorry?
I think that in Parkinson disease, the more types of cognitive neurodegeneration is all involved.
Well, that's a good question. That's a good question because we say since long time, we say that there are, and we define that there are Parkinson's disease and Alzheimer's disease. But in fact, these are not diseases. According to the classical classification in medicine, there are syndromes. What does it mean, syndromes? Syndromes, that it means that there is a Parkinson's syndromes. It means that this is a group, a number of diseases with the same manifestation, but which can be a little bit different by the causes, or by the manifestation, or by something else. And also the same for Alzheimer's disease. But what is important, that even if there is a predominated degeneration of neurons which are responsible for movement, there is the general systemic degeneration of other neurons, in Parkinson's disease, for instance, including neurons which are involved in the regulation of memory and learning. And therefore, in Parkinsonian patients, we need also the loss of cognitive function. And on the other hand, in patients with Alzheimer's disease, there is also a systemic degeneration of the neurons, including the neurons which are involved in the regulation of motor behavior. And therefore, in Alzheimer's diseases, there are also the secondary symptomatic manifestations of movement disorders. Yeah? You're quite right. Good. Now look, I showed you this slide, I think, during the first lecture. What is interesting? That according to nowadays data, the socially important diseases are presented first by cardiovascular disease, the most high incidence of patients with cardiovascular disease. The second place is occupied by oncological diseases. And the third one by neurological and mental diseases. But at all the international meetings, at least over recent 15 years, I participated in both of them. It was declared that we're waiting for a so-called neurological epidemic. Well, so it means that neurodegeneration diseases will be a global challenge of the 21st century, of our century, because of the high increase of the incidence of the number of the neurons mostly with depression, but also with neurodegenerative diseases like Parkinson's disease and Alzheimer's disease. Well, what do you think? And in fact, there is a high increase in the incidence of people with depression in Western countries and in our country, particularly between the young people. What is the reason? What is the cause? What do you mean?
The
Sorry?
awareness. So more people think about what, so more people know that their condition is a depression and not just simple sadness.
Yeah, exactly. Because it is provoked by the instability, non-stability of our life. Huh? In all countries, for instance, there is an economical crisis in the Western countries, in our country. There is a lot of problem in young people. I think that at your age, you should think always, how shall I get money to buy apartments? Yeah. I think not all of you have apartments, the old apartments, yeah? Are there somebody who has personal apartments, flat? Nobody? Well, all of you should think about that. And this is a really great problem. Well, and there are many other causes. You are quite right. This is a consequence of instability, non-stability in life, particularly in young people, because the young people, after their graduation, they can find with difficulty their place in the society. They have certain difficulties to find out a good job also. Huh? Okay. Well, so it means that that depression is affected by mostly young people at the age of 20, 30 to 35 years. Okay? And on the contrary, Parkinson's disease and Alzheimer's disease, these are diseases of aging people. Well, and there are the concrete figures for Parkinson's disease and for Alzheimer's disease. There's a lot of people in any country, very high cost for their treatment and for rehabilitation. And this is the background for the development of a global pharmaceutical background market. Look, all over the world. Well, in our country, I think this is one of the most healthy countries. But in this case, but mostly because there is not very good diagnosis. I think so. This is my personal opinion. Well, and but still, in Russia, there are 200,000 people with Parkinson's disease, and most probably there are, according to the mathematical extrapolation, there might be up to 500,000 of Parkinson's people. And Alzheimer's diseases, the number figure is much higher. This is about 1.5 million of people which suffer from Alzheimer's disease in our country. This is a great number. Well, and you remember my joke about this statue and dragon? No? Nobody remembers. Well, so certainly I will tell you that the prophylactic of Parkinson's disease is the natural prophylactic, not pharmacological prophylactic, but the natural is the training, physical training. Huh? And the prophylactic of Alzheimer's disease, this is cognitive training. Okay? But several years ago, before COVID, I participated in the International Congress in Maryland, in the United States, at a general neurological conference. It wasn't specialized in neurodegenerative diseases. And some people ask me, but could you tell me whether cognitive training, not looking at this photo, whether the cognitive training promotes the development of Alzheimer's disease? Well, I told them that, on the contrary, cognitive training prevent the development of Alzheimer's disease, but this is an exception. This is got attack, let's say, on the people who destroyed the Soviet Union. Well, so now let's prediction aside the pathogenesis of Parkinson's disease. Well, so it was 40 years ago when I'm participating in. There are two congresses all over the world which are organized annually and which are devoted mostly for neurodegenerative diseases. Well, so and the president of one of the congress, he said 1 year ago, when he gave a lecture, that 30 years ago we were absolutely happy when thinking about the pathogenesis of neurodegenerative diseases and including Parkinson's disease, because we considered that the only reason, or the only cause, the only factor which promote the development of these diseases is the loss of the one selective population of the neurons, only one of the brain. Well, so in Parkinson's disease, these dopaminergic neurons in the substantia nigra, nigrostriatal system. For Alzheimer's disease, these are neurons in the hippocampus and in the hippocampus and in the cortex. Okay, and there is another neurodegenerative disease which is provoked by the loss of dopaminergic neurons. This is hyperprolactinemia, a loss of hypothalamic dopaminergic neurons. But could you explain me why the loss of these neurons in the hypothalamic region results in the development of hyperprolactinemia? Could you recall me? Because when we discussed the role of the hypothalamus in the regulation of the whole body, we discussed this point as well. Huh? Do you remember? Why the degeneration of hypothalamic dopaminergic neurons results in the development of hyperprolactinemia. Hyperprolactinemia, it means that the decrease of the level of prolactin secretion by the hypophysis. How it happens?
Because it is the hypothalamus that controls the secretion of prolactin regulating.
But what kind of regulation? What is the neurohormone? Which is sorry?
It's inhibitory regulation.
Exactly, by dopamine. Yes, exactly. Well, so dopamine provides an inhibitory control of prolactin secretion by the hypophysis. Well, so it's naturally that under the degeneration of dopaminergic neurons and under the loss of dopamine, there is no anymore inhibitory control, and it provokes an increase of secretion of prolactin. Right? Clear? Huh? All right. Good. Okay. Let's go there. Well, so that was, I repeat once again, that 30 years ago we were absolutely happy when knowing that there are only individual populations which are responsible for the development of these diseases. And therefore, at that time, there was developed an invasive treatment for Parkinson's disease. It wasn't, yes, it was an invasive treatment. What does it mean? That look, please, there was by that time, by the end of the 80s, it has been demonstrated by a great person, a Swedish person. His name is Björklund, Professor Björklund from Lund. He has demonstrated in animal models of Parkinson's disease that the transplantation, the implantation to these animals with the degraded dopaminergic neurons, the implantation of healthy dopaminergic neurons has a 100% therapeutic effect Well, so the movement disorders of these animals, they completely disappear. So I repeat once again. He developed a model of Parkinson's disease by destroying the dopaminergic neurons of the nitrous striatal system. It provoked the development of the abnormal motor behavior in animals. But then he took healthy neurons in another animal, dopaminergic neurons, and he implanted these neurons into the brain with this destroyed dopaminergic neurons. And the movement behavior was completely improved in these Parkinsonian animals. This is clear? Clear, yeah? Okay. Well, so he was very happy, not he, but all the people. And we can see that at that time, that was at the beginning of the 90s, that we will be able to use the same technique to treat patients with Parkinson's disease. And therefore he created this Björklund. He created, so the idea was, look, this is a degeneration, these neurons, dopaminergic neurons, which are located in the substantia nigra. They send the axons to the striatum, and in this region, dopamine is released, and it controls motor behavior, okay? But in Parkinson's disease, there is a degeneration of the neurons, a loss of the neurons, and the loss of these efferents. And this is the reason, this is a cause for the development of Parkinson's disease, okay? Well, so he succeeded to improve motor behavior. So this is Professor Björklund, and this is another one. Do you recognize him?
Yes.
It's you.
That's very kind of you, because it's extremely difficult to recognize me in this photo. I think that was 1991, 92. Well, so I was in Lund, and he created a European program. For the development of clinical trials, or to try whether this technique, this approach, would be also useful for human beings, for patients. Okay. Well, and there were a number of countries, so different countries, which contributed to this program. Certainly Sweden, they were the leaders. France, Great Britain, Netherlands, Spain, Switzerland, Finland, and he invited me. At that time, I stayed in France almost for 100% of time, but still, he proposed me to create a Russian team, and they did it, and they created a Russian team, which consisted of Institute of Development. That's my institute, and we were responsible for the development of this suspension of cells, which should be implanted. Okay. Then Bordenko Neurosurgical Institute, we made the operations in patients in this institute for neurosurgery. Medical Academy of Postgraduate Education, the neurologists were recruited from this medical academy. Scientific Center for Obstetrics, Gynecology and Perinatal, because at that time we were obliged to take these neurons, healthy neurons, from the embryos of humans or women, and therefore in the same day before the operation, we had to get at least 5 embryos during the abortion, and there was an abortion under the control by ultrasound. Well, so that was made in Scientific Center of Obstetrics. Nowadays, it's completely impossible for ethical reasons. But in the 90s, not only in other countries, but also in other countries, that was possible. Still, it was possible. Institute of Transplantation and Artificial Organs, because we had to make immunosuppression just to prevent the rejection of these neurons from the brain, implanted neurons from the brain. And the Institute of the Normal Physiology, I was here to the laboratory also there. Well, and this is, in fact, this is a, what, let's say, schematic representation of the operational, of the cipher. So in the beginning, we had to obtain embers at age, as far as I remember, about 12 weeks. 12 weeks, because after that it was restricted to make abortion at 12 weeks. Huh? Then we dissected, under the microscope certainly, we dissected this small region, midbrain, which contains progenitor or dopamine, newborn dopaminergic neurons themselves. Then, that's also me. Then I prepared a suspension of these cells. That was a piece of tissue, huh? And I prepared a suspension of this. I dissociated the cells. And then I transferred this suspension to a neurosurgeon, and this neurosurgeon, he used so-called stereotactic operation. He made a small hole in the brain, okay, and he inserted very precisely this cannula with this suspension into the striatum of patient, and then he injected this suspension into the brain. That was a typical operation that was made in all the countries. Okay. And then it has been demonstrated, first, that the motor behavior was improved in patients, but not in all of them, only in 30%. And that was not a long-term process. That was an improvement for maximum for an hour, a year, maximum for a year. Huh? And then it disappeared. You could look at this graphic, at this representation of the increase of the time of this movement. Okay, after transplantation. Well, so that was before the operation. It took much time to make this toning, but after the operation, the time was shorter, was significantly shorter. Well, then it has been demonstrated that these implanted neurons, they survived in the brain of the recipient, of the patient, and they express these are neurons which are stained for tyrosine hydroxylase, for the first enzyme of dopamine synthesis. And it has been also demonstrated in the positron emission tomography, with the positron emission tomography, that there is an increase of dopamine synthesis after the implantation. Well, look please, this is before implantation. The Parkinson's disease is developed asymmetrically. On the one hand, the damage is much higher than on the other hand, on the other side. Look, please. This is mostly normal structure, and this is a structure with the loss of dopamine synthesis. Okay, on the right and on the left. And then after the implantation of the neurons to this region, there is a high level of dopamine synthesis. Look, even higher than on the other side. Well, so we have demonstrated finally that these neurons, well, so I should mention that it was impossible to implant the neurons which were taken from the brain of adult persons. They could not survive in the brain of the recipient. They were obliged to use only embryonic neurons. And embryonic neurons, they are well differentiated in the brain of the recipient, They express the specific dopaminergic phenotype, and they synthesize a lot of dopamine. But finally, altogether, all the teams of all the countries, we collected for several years, we collected 200 patient cases. And when evaluating the whole populations of these separated patients, we came to the conclusion that directed dopaminergic neurons survive up to 10 years and reinnervate the striatal patients. This is very good. This is excellent conclusion. Patients show improvement of therapeutic value. This is good, but this is bad. But the symptomatic relief was variable, temporal, and incomplete. And this is really bad. Neurotransplant, and the final conclusion, so because of this negative results, was as follows. Neurotransplantation cannot yet be recommended for treatment of patients. This conclusion was made at the beginning of 2000. Okay? Well, and but still there were very enthusiastic people, particularly of your age, because they defended, they made their theses in PhD theses with this neurotransplantation. And they were really a very great enthusiasts of this direction. Well, so they decided to continue this study by improving the technique or improving the materials which is implanted. What were the ideas? What were the objectives at that time? The promotion of dopaminergic integration of the striatum by increasing the number of implants. Well, so I told you that we implanted the materials which were taken from 3 to 5 embryos, human embryos. But they considered to use more embryos, 10 embryos, for instance. This is the first objective. Then, to protect or correct the dopaminergic neurons from death by using antioxidants, these are neuroprotectors, growth factors, and caspase inhibitors. Caspase inhibitors, they inhibit what they inhibit? Caspase inhibit apoptosis. Apoptosis. Good. Well, and then xenotransplantation of dopaminergic neurons, mostly from teeth, because even at that time, it was allowed all over the world to use abortion material, but still the ethical problems arose even at that time. And therefore they considered to replace human materials by pig materials. Because according to immunological processes, the human being is very close to pigs. I'm sorry to say that, but some people, they're close even for other factors. Well, good. Then, development of alternative sources of cells suitable for transplantation. At that time, there was an idea to use genetically engineered neural and non-neural cells. What does it mean? It means that there was an idea to transfect any cells, a neuron or muscle cells or fibroblast or any cell, with the enzymes of dopamine synthesis, okay? And to use them for implantation. That encapsulates cell lines. What does it mean, encapsulate of cell lines? Certainly, there was always a risk of immunological rejection of implanted cells. And therefore, they proposed to put these cells, implanted cells, into a capsule with a small holes which were permeable for neurotransmitters and very small molecules, but which were not probable for large molecules of gamma globulin, which reject the neurons. You understand what I mean? Huh? Everybody?
No, no, please,
Nobody?
come on.
No?
Yes. Yeah, go back.
Okay.
Thank you.
Okay, good. Well, so that was another approach. And then stem cells, to use stem cells themselves, and genetically engineered stem cells. Well, so stem cells with the transfected crucial genes, mostly for enzymes. Well, but what is the result? 30 years has passed. Have passed. 30 years. But with any population of implanted cells, we're coming to the same conclusions as in the program of Ugrumov. There's excellent therapeutic effect in animal models and no effect in patients. Well, but still, these cell technologies, they continue to be developed all over the world with the hope to succeed finally. But I told you I told you earlier, I'm not sure if you remember it, that I have an idea, personal idea. So I left this program at the beginning of 2000 after this conclusion, and that was also the case for Björklund, who was a founder of this direction. Well, so my idea is the creation that there is a taboo, sort of biological restriction. What does it mean? The creation of any dopamine pump. Well, so the idea of Björklund finally, when he developed an experimental model in mice, Parkinson's disease, the idea was that we should, that after the degradation of dopaminergic neurons, we could insert into the striatum any pump for dopamine. Any, huh? Well, so, and these neurons, implanted neurons, they were considered as a source, as a pump for the release of dopamine. Okay? But it means that in this case, this dopamine could realize only volume neurotransmission, but not synaptic neurotransmission. Okay? Well, and look, the creation of the dopamine pump is in the brain would not be beneficial for the improvement of motor behavior in humans. Real success might be probably achieved under the, I think it's under the reconstruction of the synaptic connectivity neuronal networks that is almost impossible nowadays. Well, so my idea was at that time and nowadays that in mice there is volume neurotransmission predominates. And therefore, if we use cells which secrete directly into the extracellular space dopamine, so we could improve, in mice, we could improve motor behavior. In human being, the most important is more strict regulation, synaptic regulation. Why a point like synaptic regulation? And any neuron, dopaminergic neurons, in the substantial negative, it receives from 10 to 10,000 of pixels, which synaptically control the functional activity of these neurons. And I believe that at least in the near 50 years, nobody will be able to reproduce these synaptic connections between one dopaminergic neuron, any dopaminergic neurons, and 20,000 of other neurons. It is impossible because I gave you a good example. We don't know how we could construct the certain synaptic connections between the neurons. For instance, if we take, I mentioned already one day, if we take a brain region, the same brain region, 100 times, huh? if we dissociate this brain region into tissue culture, dissociate into individual cells, and put them into tissue culture. Always there will be developed synaptic interrelations, but always different. Always different. Well, so we are not capable nowadays to reproduce a certain synaptic relations between certain neurons. We don't know how to do it. There is a biological restriction, and therefore I consider it as a taboo. And this is the case because I don't know, all these neurons were used for the transplantation with no positive effect. Well, and now I think it's time to break. And before, I would like to propose you for the next seminar these papers. I think we will select four. You will give four presentations like today, okay? Please, could you tell me who is interested in preparing these presentations? Are there volunteers? Okay, I will send this list to Alisa, and Alisa will distribute between you, and you will select, okay? Alisa. Well, so there are papers on neurodegenerative diseases because we continue. Our lecture would be devoted to neurodegenerative diseases. Well, so different aspects of Parkinson's disease, and mostly Parkinson's disease and Alzheimer's disease. Well, so there are four papers on this topic. Good. Now, I think you will have a break. For how many? For 20 minutes? For 20 minutes? Okay, in 20 minutes we will